Hello everyone, hope you all are doing well. Today I am here with the fourth lecture series in the topic photosynthesis. So earlier we have discussed about all the basic pigments, process, how it works and everything, and today we will just get inside the process. So today we will discuss about the light reaction, or specifically the non-cyclic pathway of light reaction, which is the major pathway in photosynthesis in green plants. So let's get started. So today we will again read about the introduction of light reaction in photosynthesis. Earlier previous class we have also read that, but today just for the recapitulation, so that it would be easy to understand about the non-cyclic photophosphorylation, we will study it in a bit details only. Okay, and then we will study about non-cyclic photophosphorylation. So let's start. Now, what is light reaction? We all have studied about the light reactions, the reactions which occur in the presence of light in the thylakoid of chloroplast in green plants. Okay, so these are simply termed as light reactions. Definitely they are light dependent. And what is the output of these reactions? These reactions, what are the types of reactions present happens in this system? Oxidation of water, reduction of NADP plus, and ATP synthesis. Okay, ATP synthesis is the major process or the major thing happening here, for which this process has got the name photophosphorylation. Okay, so ATP synthesis triggers its name to be photophosphorylation. Now all of these processes may occur in two ways, one in non-cyclic way and the other in cyclic way. From the name only you can differentiate. In non-cyclic way there won't be any cycle, that is there would be no fueling back of electrons to the original source, and in the cyclic pathway there will be a fueling back of electrons to the original source. But here, to your information, I will tell you that the non-cyclic photophosphorylation is the major kind of pathway. Cyclic photophosphorylation also occurs, but non-cyclic pathway is the major one and very important for whatever examination you prepare. Okay, so let's study non-cyclic electron flow in photophosphorylation. Now, what do you understand by photophosphorylation? Photo means light and phosphorylation means addition of phosphate. So, the addition of phosphate to ADP, inorganic phosphate, creates ATP. So, the particular process is termed as photophosphorylation. Now, let us discuss the points whatever is wrote in the slide. It is a light induced electron transport from water, that is photolysis of water occurs with the release of electrons, a pair of electron, and when to which the electrons are transferred, it is transferred to NADP plus, which then gets reduced to NADPH. We will study it. Okay, and what is evolved? Continuous evolution of oxygen is seen in this process. Then PS2 and PS1 are both involved in case of non-cyclic photophosphorylation. Now reaction stimulates from the reaction center in PS2, from where the electrons are carried to PS1 and lastly fed to NADP plus. Now for the amateurs I am telling that the reaction doesn't start from PS1 and then it goes to PS2. Okay, this is not like any conventional process you will study, like you will tell ki PS1 and then PS2. No, for amateurs only. The people you know it very rightly that the process continues from PS2 and then it goes to PS1. Now what is PS2? You all know it is P680 and PS1 is P700. Okay, and the reaction proceeds through the Z scheme. You will see in the next slide that the reaction process, how is it or why is it termed as Z scheme. Although to us it will appear like an N, but scientists may have seen it from a different perspective, like the reverse of N, and that's why, like the rotated version of N, and so they have named it as Z scheme. Okay, let's start. Now see, here I have given you a clear illustration of the molecules involved in the Z scheme. You can take a screenshot of this image and save it in your phone, and you can also draw it down Okay, this picture is very necessary and is the total gist of whatever is taught in this slide. So I'll discuss a bit from the beginning. So at first let me tell you that the reaction starts from P680. Okay, the reaction starts from P680, that is PS2. Here we have the reaction center in which the main part is chlorophyll A along with other accessory pigments. Now what happens when the electron is released? Like the electron is released and it is getting excited. First the sunlight falls and then there is a release of electrons through the photolysis process, which I will discuss after discussing the cycle. Okay, then it would be totally clear. Like the first you assume that the electrons are released, and as because the electrons are released, the P680 system gets excited, and due to excitation it's what happens in excited state, the particular complex is not stable, so it has to return to its ground state, and in order to return to its ground state it transfers its electron to the further molecules. Okay, so what molecules they have in this process? So they transfer the electrons to pheophytin. Whom? Pheophytin. Now what is pheophytin? Do you people know what is pheophytin? It is a type of chlorophyll molecule only. You will study it in details as I proceed through the slides. Okay, it is a type of chlorophyll molecule only. So now what pheophytin does? Pheophytin transfers these pair of electrons to QA and QB, which are plastoquinones. Okay, and from the plastoquinones these electrons further travels through the cytochrome B6F complex, and via this B6F complex it travels to plastocyanin. Okay, so this is the total channeling from P680, then pheophytin, then through QA and QB to cytochrome B6 complex, and then to PC, that is plastocyanin. Understand this part. Now in this particular part, in the region where there is cytochrome B6F complex, let me tell you, when the electron moves through this region, there is a release of lot amount of energy. There is a release of lots of amount of energy, which helps us in combining ATP plus inorganic phosphate, which helps not us like plants to combine ATP and PI to form the ATP. So here the ATP formation takes place. I will make a different slide about detailed study of ATP synthesis in photophosphorylation process. For now you just know that this is the particular region where ATP is synthesized. So up till now what we have read, that there is the transfer of electron, and in the cytochrome B6F complex region, particularly near that region ATP is synthesized. Now let's move on. The electrons from the plastocyanin, what happens? They further move to PS700, which is PS1 system. Now PS700 gets excited, so it has to return to its ground state. So in order to return to its ground state, it further transfers its electron to the A0 complex, which is a part of P700 only, and then it transfers its electron to A1, which is a quinone. Okay, the next acceptor in this process is the quinone. And then you have few iron sulfur proteins. I have not named the proteins here. You may write it down. Those are FX, FA, FB. 3 kinds of protein. More are there, but these 3 are very important. So there is a series of iron sulfur proteins through which these electrons pass, like they are accepted from one particular electron and then passed or donated to the other one. And in this way the electrons travel through the iron sulfur proteins further to ferredoxin, which is written here as FD. Okay, and then this is the last step, like they are transferred to the ferredoxin, and after that what happens? The NADP plus molecule is reduced to NADPH by the aid of ferredoxin NADP reductase enzyme, which is present there. So what happens now? How is it reduced, or what happens? Now we will come to the photolysis of water thing. So as sunlight falls, the water molecule breaks. The water molecule breaks into liberate one molecule, like if we break one molecule of water, then we will have one nascent oxygen, two electrons and two protons. But here I have shown two molecules of water so that I can show a single molecule of oxygen, okay, for better understanding. So two molecules of water are seen here, where there is the photolysis of water taking place to produce one molecule of oxygen, 4 electron and 4 hydrogen. And you know what happens now? These electrons serve as electron chamber for the P680 system. So I told you at the end of the process you will learn the where from the electrons come. So the electrons are fueled from here to P680. Now these electrons cycle through this process and further they come to the last point where it is coming to the ferredoxin, and then with the aid of NADP reductase enzyme, which is present in the outer membrane of the thylakoid. This whole process is taking place in the thylakoid. The ATP formation and other stuffs are taking place in the inner membrane of thylakoid, while this FNR enzyme, it is present in the outer membrane of the thylakoid, okay. So in the outer membrane of the thylakoid it is present, where this NADP plus is reduced to NADPH with the aid of that enzyme, and the protons we gained from photolysis of water. So the reaction would be NADP plus the protons, which give us NADPH, okay, by the aid of the enzyme ferredoxin NADP reductase. So it is clear. So let me tell you the gist again. So in this, so you can see the scheme, it is a Z scheme. If you reverse or rotate your phone a bit, then you will see that the process is a Z scheme process. So in this total process, what happens? There is photolysis of water due to sunlight, which gives us certain electrons which are fueled through the PS2 and then through the PS1 to produce NADPH molecules, and in NADPH molecules in the outer membrane of the thylakoid and ATP molecules in the inner membrane of the thylakoid. So there are two products of this particular reaction taking place, which are ATP and NADPH, and one more byproduct we have in this process, which is directly the oxygen, which we get from the photolysis of water, which is the oxygen. Oxygen is the byproduct, and the end products of the reaction are ATP and NADPH. The complex for photolysis of water is present also in the inner membrane of thylakoid, okay. So oxygen is also produced in the lumen of the thylakoid. Only what? NADPH2 is produced in the outer membrane or in the stroma actually. So this is the gist of the process. I have explained to you the details, and here I have mentioned the abbreviations. You can check it in the diagram. While you draw it in your copy now, write the abbreviations down below, okay, so that it would be easy for you to just take an eye on it. Go through the abbreviations once, and then we have the process. Whatever I have discussed here, I have written it in the gist. I will read it once for you all, okay. Excited PS2 reaction center chlorophyll transfers the electron to pheophytin. Pheophytin supplies the electron pairs to plastoquinones, which are QA and QB. Cytochrome B6F complex that accepts the electron pair from plastoquinone and transfers it to plastocyanin. Then the electron pair moves to PS1, which is P700. A part of it, which is A0, transfers this electron to A1, which is the quinone center, and then it reaches to ground state itself. Then a series of iron sulfur proteins, which I have already mentioned previously, I have not mentioned in the slide, you can note it down, FX, FA and FB. These three proteins helps in transferring of electrons to soluble ferredoxin, which further with the aid of FNR enzyme, which is ferredoxin NADP reductase, converts NADP plus to NADPH. Once again I will repeat this thing, that in this process the byproduct is O2, oxygen, and the end products of this process are ATP and NADPH2. NADPH2 is produced in the stroma region, while the oxygen and the ATP are produced in the lumen or the inner side of the thylakoid membrane. So this is the gist of overall non-cyclic process. I will teach you the ATP synthesis in details after I teach you the cyclic photophosphorylation. It is also a small topic. I will come up with it in the next slide. Okay. So thank you, children. I hope you all are studying well.